Ethylene is the only plant hormone that is gaseous in nature. It produces naturally by higher plants and is able to diffuse readily via intercellular spaces throughout the entire plant body. Ethylene is basically involved in plant responses to environmental stresses such as flooding stress, such as drought stress, and in response to infections, wounding, or mechanical pressures. It also influences a wide range of developmental processes include shoot elongation, flowering, seed germination, fruit ripening, and leaf abscene. If we talk about ethylene actions, ethylene is basically involved in signal transduction. Several transmembrane proteins have been identified that bind to the ethylene at the cell surface and functions as signal transduction. Ethylene has major role in fruit ripening. Under natural conditions, fruit undergo a series of changes include change in color, decline in the organic acid content, and increase in sugar content. In many fruits, these metabolic processes often coincide with a period of increased respiration, the respiratory climacterics. During the climacterics, there is also dramatic increase in ethylene production. Basically, ethylene initiates the climacterics in a number of fruits and is used commercially to ripen tomatoes, avocados, melon fruits, etc. Here, this is the ripening of the fruit banana in presence of ethylene. Ethylene is also involved in shoot growth. Applied ethylene has the capacity to influence the shoot growth. Applications of ethylene to dark ground seedlings can cause reduced elongation of the stem, bending of the stem, and swelling of the epicotyle or hypocotyle. They are also involved in flowering. The ability of ethylene to affect the flowering in pineapple has important commercial applications. Ethylene also promotes the flower senescence, aging in plants such as petunia and peas, etc. Thigmomorphogenesis, the change in growth response, a growth form in response to mechanical stimulus such as starch, that is also regulated by ethylene. Next is the brasinosteroids. Brasinosteroids are plant steroid hormones that have similar structure to animal steroid hormones. They have multiple developmental effects on plants, including promotion of cell elongation, cell division, and xylem differentiation. Brasinosteroids are basically deficient mutants that exhibit dramatic growth defects, including dwarfism, reduced apical dominance, and male fertility. Brasinosteroids switch on specific genes by inactivating a protein that otherwise indirectly blocks transpiration of those genes, transcription of those genes, leaf abscene.